Let me bring our first moderator, Lauren Whisper from the University of British Columbia, Canada.
So welcome, everyone. Before I introduce the first speaker, I wanted to thank Vivian for spearheading the Climate Change Symposium. She's really done a wonderful job. And also Jeremy and Heather for helping with many useful suggestions. And of course, Amelia for helping with the organizational details. So I'm very pleased to introduce our first featured speaker. It's early here. Dr. Jill Anderson from the University of Georgia. Jill, I think as probably many of you know, examines the ecological and evolutionary consequences of global change for natural plant populations. And I think her work's been super high profile and impactful. And today she'll be talking about local adaptation in a rapidly changing climate. Jill.
Thank you so much, Lauren. Let me share my screen and then I'll get started. All right. Perfect. Okay, as Lauren mentioned, I am a plant evolutionary ecologist, and I'm very interested in the constraints on adaptation in natural populations. Species evolve in very complex environments where they're subject to variation in abiotic and biotic conditions that can change across the landscape. And divergent selection can favor the evolution of local adaptation, which is a pattern that has evolved in numerous species across the tree of life. Reciprocal transplant experiments have been a really powerful approach for detecting genotype by environment interactions in fitness, and they often reveal that local genotypes have elevated fitness in their home location and depressed fitness in the contrasting environments. However, despite how common local adaptation is, it's not ubiquitous. And that leaves us with the really intriguing task of trying to investigate the processes that constrain local adaptation and drive fitness in natural landscapes. In addition, these reciprocal transplant experiments have often focused on really contrasting environments where, when we know that species are distributed across environmental gradients, where conditions might change subtly and different agents of selection may be more or less important in different portions of the range. So this is a species distribution model for my focal species, Bucra stricta, which was put together by my former graduate student, Derek Denny. And this really allows me to highlight the two main questions that I'd like to discuss today. So first, the question is, how do different agents of selection contribute to the evolution of local adaptation, especially in the context of climate change? Because one might imagine if local populations are adapted to non-climatic factors such as soil differences, soil nutrient differences, then change in the climate may not have pronounced effects on fitness and population persistence. Instead, if these local populations are strongly adapted to contemporary climates, that could very much disrupt local adaptation going forward. So I'd also like to address the question of whether in situ evolution or gene flow could rescue populations confronting climate change. And over the past 10 or 15 years, I've really focused on elevational gradients because there's this beautiful natural laboratory for examining systems in which the climate changes gradually, but really actually quite strongly as you go up in elevation. And oftentimes you see that multiple climatic factors are changing across these elevations. So here we see that this is Gothic Mountain around the Rocky Mountain Biological Lab, and we see that temperatures decline and the timing of snow melt also is delayed in high elevation populations. Now, there are other agents of selection that also differ across this gradient, which allows us to try to disentangle these various agents. And finally, this is a really interesting system because one might imagine that strong gene flow between populations at different elevations could potentially constrain adaptive responses to climate. So I focused on Bucra stricta, which is this really beautiful species that's common throughout undisturbed habitats in the Western United States. I love this species because it exists across a broad elevational gradient and a broad latitudinal range. And there's biogeographic evidence to suggest that it's been able to shift its distribution in response to geological periods of climate change. So for example, the expansion and contraction of the Pleistocene glaciers. It's a short-lived perennial, which is primarily selfing, and that means it's an extremely experimentally tractable system. I just wanted to show you a little bit of the distribution of Bucra. These are the locality data that Derek used in that species distribution model that I showed up front. And my work has really focused in this region in Colorado around the Rocky Mountain Biological Lab. So I'd like to highlight that this is the sovereign territory of the Ute people who were really driven from this land during the gold rush. So in 2013, I first established 5 common gardens arrayed across an elevational gradient in this general region. And since then, we have transplanted large numbers of Bucra stricta individuals at both the seed and the juvenile level into these common gardens. Since Bucra is a short-lived perennial, we use both seeds and juvenile rosettes so that we can get adult traits from these rosettes that we transplant. It typically takes seeds 1 to 3 or 4 years before they will begin reproducing after you transplant them. We've used many families that were sourced from diverse populations distributed across this entire gradient. And as a perennial, Bucra, like many species, has a really complex life history. The seeds germinate in early spring. They grow as juvenile rosettes. Here I indicate for the first year, but really it's for the first 3 to 4 years. They overwinter, and then they use the vernalization cues they gather from wintering to flower in subsequent years. So the next spring, they might begin to bolt, which you can see here. Then they flower. They produce seeds. Some of those seeds enter the seed bank and can germinate in subsequent years, while the adult plants are overwinter underneath the snow. So first, I'd like to look at local adaptation across 3 really important fitness components in the system. And I've followed a provenance trial approach, so I just want to orient you to the figures that you'll see. We're first looking at germination success. Germination is often an underutilized fitness component because it's really hard to transplant seeds and to follow germination success of those and get early fitness components from those transplanted seeds. But here we're looking at germination success, and we'll look at 2 other fitness components as a function of the elevation of origin of all of the accessions that we've transplanted into these gardens. And I'll show you the curves for all 5 gardens. And essentially the prediction is that we should see elevated fitness for genomes or for genotypes that evolved in environments similar to the elevation of the garden. So here, at this lowest elevation location, we see that low elevation genotypes have a fitness advantage over their higher elevation counterparts, and that's consistent with our expectations for local adaptation. Similarly, at our mid-elevation sites, we see that germination peaks for genotypes that evolved in those environments. You don't see perfect local adaptation across the range. At this higher elevation site, the patterns are somewhat contrary to predictions, and there may be some evidence of local maladaptation under contemporary climates. But we regain local adaptation in our highest elevation garden, where high elevation genotypes have elevated fitness. We can also examine these fitness curves in response for survival of juvenile rosettes. And again, we see patterns that are strongly consistent across most of these gardens with local adaptation, such that low elevation genotypes do best in low elevation environments, and high elevation genotypes have the highest fitness in high elevation locations. Finally, I'd just like to show you some fecundity data. So this is essentially seed set. And again, you see patterns that are mostly concordant with local adaptation. But when we examine all 3 of these together, I'd just like to make 2 points. One is that there is evidence of local adaptation to elevation, but this local adaptation varies across space, across these different gardens. as well as across life history and fitness components, which just highlights to me that it's really crucial for us to examine these patterns in multiple locations using multiple fitness components. But if we want to make predictions about climate change, we can't simply examine local adaptation under contemporary climates. We, in fact, need to simulate future climates to examine how fitness varies as a function of climatic factors that are changing. And we all know that temperature is changing worldwide. So it's relatively straightforward to simulate elevated temperatures through open top chambers. This is a beautiful picture of my highest elevation garden, and you can see the open top chambers within the garden which we've placed over some of the experimental blocks. But as many of you already know, in high latitude and high elevation locations, another driver is very important in determining fitness and vital rates, as well as phenology and other life history functions, and that is the timing of snowmelt. The timing of snowmelt is shifting rapidly with climate change, and it's shifting rapidly in almost all locations where snow is a dominant player in the ecosystem. So this is just looking at shifts in snowpack timing or snowmelt timing, shifts in snowpack across the Western United States. This is the Rocky Mountain Biological Lab where I've been conducting my research, and you'll see that most localities have received reduced snowpack in the last 40 years or so. This is driven by several factors. One is that warming winter temperatures are causing precipitation to fall more as rain than as snow. So we're seeing that the snowbank isn't building up to the same extent as it used to. And then warming spring temperatures are causing the snow that does exist to melt out earlier. That also advances the dry period between snowmelt and monsoonal conditions. So this advancing snowpack has really pronounced effects on the entire ecological community. So within these gardens, we have also been manipulating the timing of snowmelt. So this is in our second to lowest elevation garden, immediately before we shoveled snow for the snow manipulation treatment. And then this is 10 days later, where you can see that the snow removal blocks have already been exposed to the environment. So early snowmelt shifts phenology. This is germination and flowering phenology, as you'll hear later on. It exposes plants to frosts that they wouldn't have experienced when insulated by this blanket of snow, and it decreases soil moisture. So now we can examine fitness across the lifespan in response to these key agents of selection that are shifting rapidly with climate change. And I showed you this life cycle before, but Megan DeMarsh and I have recently formalized this life cycle into an integral projection model, where we explicitly model vital rates as a function of plant size, as well as these two climate drivers. I won't show you all of the vital rates, but I wanted to highlight two. This first one looks at germination success. So here we're modeling germination success as a function of snowmelt timing by source elevation across all 5 gardens simultaneously. And you can see in this component of fitness that an intermediate snowmelt timing is really favored for almost all of the genotypes in the experiment. And that's somewhat unusual given how prominent local adaptation at this particular vital rate was when we examine elevation. So this suggests there may be other drivers of local adaptation to elevation for germination. But we see patterns that are highly consistent with local adaptation to snowmelt when we look at other vital rates. So this is looking at seedling survival. And here, the high elevation genotypes do best under delayed snowmelt, which reflects their historical climate. And lower elevation genotypes have elevated fitness when snowmelt is earlier. So after integrating across all of these vital rates, we can actually look at the climate niche. So we can model for each genotype in the experiment its response to snowmelt timing or growing season temperature. So this is lambda. As you know, a lambda of 1 means a population is stable. Above 1 means it's growing, and below 1 means it's contracting. And from these curves for each genotype, we can extract the climatic conditions that optimize lambda. So basically the optimal snowmelt timing for each genotype, which would just be the peak of these curves. And then we can examine whether there is in fact local adaptation to these two climate drivers. So here are our predictions. These are elevational gradients in the timing of snowmelt and growing season temperature across the localities that we've used as source populations. And we do see evidence for local adaptation to climates that vary across elevation. High elevation genotypes are adapted to delayed snowmelt. Low elevation genotypes are adapted to earlier snowmelt. And we see a pattern in the growing season temperature optima that really reflect this elevational gradient quite nicely. So we can move on from there to examining questions about whether climate change might disrupt local adaptation. So I'll be showing you fitness curves for all 5 of our experimental gardens. And on each curve, you'll look at 4 different climate scenarios, a pre-industrial scenario, a contemporary scenario, and 2 end-of-century climate scenarios. And in our lowest elevation garden, where populations truly are contracting, the natural population is contracting in this location, you'll see that we're only truly able to capture values, lambdas of 1, for these pre-industrial climates. And under pre-industrial climates, we see patterns that are highly suggestive of local adaptation. We can also look at the optimal source elevation. So up here, the horizontal black line indicates a lambda of 1, and here the horizontal black line indicates the elevation of that garden. The pattern of disruption of local adaptation becomes more intense as you go up in elevation. So in this mid-elevation location, the local populations are pretty well adapted to contemporary and pre-industrial climates, but we see a shift toward lower elevation genomes being favored in future climates. And that shift becomes even more pronounced as you walk your way up these elevational gradients. We also, in addition to this common garden experiment, we conducted a reciprocal transplant experiment using more local accessions from all 5 of these gardens, so we can examine the climatic niches of multiple individuals that evolved in the same environment. And what we notice here is that there's relatively limited potential for evolutionary rescue to save these populations under future climates. So these are the climatic niches, which are often asymmetrical. Here we're looking at a principal component that combines snowmelt timing and growing season temperature. So negative values reflect early snowmelt and warm temperatures. Positive values reflect delayed snowmelt and cool temperatures. And the stars on the x-axis indicate different climate scenarios. But what you can see is that our genotypic variance in lambda is significantly higher under pre-industrial and contemporary climates than it is in any future climate scenario. So if local populations don't have sufficient genetic variation to adapt to climate change, we wondered whether gene flow might rescue local populations. So here we're looking at the minimum gene flow that's necessary for populations to persist under all 4 of these these climate scenarios that we've modeled. And negative values would reflect downslope migration, whereas positive values reflect upslope migration. So you can see that under contemporary and green and future climate scenarios, we're projecting that those populations will only persist with a relatively significant upslope migration. So we then teamed up with James Santangelo and my former graduate student, Derek Denny, to actually model the rate of gene flow using low coverage genomic sequence data. We came up with 3 transects across elevational gradients in low elevation, mid elevation, and alpine populations. And what I'd like you to focus on here, these data come from, or these models come from James's models of gene flow. I'd like you to focus on these more recent estimates of gene flow. And the downslope gene flow is in blue, upslope gene flow is in red. And in all of these more recent cases, we see very limited evidence that there could be upslope migration. So natural upslope migration is not occurring readily in contemporary environments. Finally, Derek used the species distribution model approach to project populations under 2 different climate scenarios across the range, and we see large scale expectations of contraction in low elevation and low latitude populations, and potentially some expansion into higher latitude and higher elevation populations. But I like the contrast of the integral projection models, which take local adaptation into account, but assume no gene flow, and the species distribution models, which assume no local adaptation, all populations have the same climatic niche, but that gene flow is unrestricted. And we did some formal comparisons of the integral projection models and the species distribution models, but I don't have time to go into them. I'm happy to talk to you about them later. So to summarize, we find that climate change does shift fitness landscapes, and that does disrupt local adaptation. That genotypic variation in climate tolerance declines under projected climate. So at least in this system, there's limited potential for in situ evolutionary rescue. Large-scale population contractions, however, could be minimized through assisted gene flow protocols. So with that, I'd like to thank you for your time and attention.
Jill, thanks for the wonderful opening talk. Because of the canceled speaker later in this session, we can take a couple of questions, and then any additional questions we'll take in the Q&A session. There have been a couple questions put on the chat. I can read them to you if you wish, or you can look at the chat yourself. But here's the first one. Sorry if I missed this in your presentation. But can you comment on how adaptive plasticity would change if we move the seeds generated from a lower site and planted them on at a higher site, at a higher altitude?
Yeah, so that's a great question. This talk wasn't specifically about adaptive plasticity. Obviously, seeds moved around and plants moved around are expressing different traits in different localities. We have a whole large data set on a variety of phenological and functional traits that I didn't have time to get into here, but that is actually the next paper that I'm really excited about looking at is how the extent to which climate change may alter selection on plasticity and whether plasticity could enable populations to persist. But plasticity is inherent in these models because individuals transplanted into different locations are expressing different traits. It just wasn't necessarily a part of the talk that I gave today.
Thanks, Jill. I should say that these questions are started with great talk and wonderful slides and so forth. I didn't mention that, but just so another question was, did you mention which vital rates contributed most to lambda in this system?
No, I didn't mention that. Megan Demarche has done sensitivity and elasticity analyses with the vital rate models or with the integral projection models that we've looked at. And it's not, it is relatively complex and it is not a simple answer. And the answer to that question depends on where you are in along the elevational gradient. So as an example, the low elevation populations are really sensitive to seed dormancy, so seed production and seed dormancy, and that's primarily because there's very little seed germination. So seeds have a greater chance of staying alive in the seed bank than they do of germinating and moving forward as plants. And in higher elevation populations, it has to do with fecundity of large plants, but it's a little bit more complex than that, and I'm happy to answer in more detail later.
I'm going to ask one more question from the list, and then we'll go to the next speaker. There'll be a second chance to ask questions in our question and answer session after the next speaker. So this question comes from Holly Prendeville. I actually haven't been mentioning the names here, but what recommendations do you have for land managers to implement the findings of your research to their land management practices and decisions?
Yeah, that's a great question. I strongly believe in the assisted gene flow framework that Sally Aitken and others have discussed, and I think that it's a really powerful framework. One issue is, how do you decide which populations or which species to implement assisted migration for? So I think additional approaches involve conservation of land that could serve as corridors for populations migrating naturally.
Thanks so much, Jill. As I mentioned previously, there'll be a time in the question and answer session to ask additional questions.